Hello everyone, welcome to another video on GATE BT important topics. Today in this video, I'm going to talk about the topic probability of restriction site, a must-do topic by all GATE BT aspirants. I'm sure while going through previous year GATE BT question papers, you would have come across numerical answer type questions on probability of finding a restriction site, 6 base pair or 4 base pair in a genome, right? These questions are very scoring and they carry 2 marks. And this is the reason this particular topic you must prepare for the exam. So today in this video, I'm going to talk about the basic concepts which are related to this topic. Also, I will be telling you some strategies of solving the questions based on probability of finding a restriction site in the genome. So let's get started. Let's first know about restriction enzymes. I'm sure you all would have heard about this term, restriction enzymes, right? So what are restriction enzymes? Restriction enzymes, they belong to large class of enzymes called as nucleases. So question arises, what are nucleases? Well, nucleases are the enzymes that degrade DNA molecule by breaking the phosphodiester bond that links one nucleotide to the another, that links one nucleotide to the next in a DNA strand. So like for example, here you can see, let's consider DNA to be a string of beads. So here these beads, they represents the nucleotide, and I'm sure you know that nucleotides are the monomeric unit of DNA, and nucleotides comprises of 3 main components, that is deoxyribose sugar in case of DNA, phosphate group and nitrogenous bases. So one nucleotide is connected to another with the help of the phosphodiester bond. So one nucleotide connected to another in the same strand with the help of phosphodiester bond, right? Now in the DNA, sugar phosphate remains the same in a nucleotide, right? In every nucleotide, sugar and phosphate backbone remains the same. It's just that the nitrogenous bases which are present, they are different, right? So let's say here you have adenine. So on the complementary strand, you have the thymine because DNA is double stranded. If guanine is here, then cytosine will be present on the complementary strand. And adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds, right? So the nuclease enzyme, it breaks this phosphodiester bond. Now, question arises, which phosphodiester bond it breaks? Within the DNA molecule or near to or towards the terminal region. So basically, exonucleases, like nuclease are of 2 kinds, exonuclease and endonuclease. So exonuclease, these are the enzymes, the nucleases, which remove the nucleotides one at a time from the end of the DNA molecule, as you can see here. So this phosphodiester bond is broken, and you can see that the nucleotides are being removed one at a time from the end of the DNA molecule. However, endonucleases, endo means within. So these are the nucleases which break internal phosphodiester bond within a DNA molecule. So here you can see that from here, this phosphodiester bond is broken. So you can see the fragments generated. Further, if you break the phosphodiester bond, this enzyme breaks the phosphodiester bond, so further fragments are generated, right? So when we talk about restriction enzymes, they are endonucleases. They are restriction endonucleases, and they're popularly known as molecular scissors. So just like you need a scissor for cutting a cloth or a paper, similarly to cut the DNA molecule, we need the restriction enzymes, or also called as restriction endonucleases. So popularly they are called as molecular scissors. But the very interesting fact about these restriction enzymes are that it doesn't cut randomly. It recognizes specific base pair sequence in the DNA, and then it makes a cut. So I'm sure when you study about gene cloning and recombinant DNA technology, so basically you know that in gene cloning or recombinant DNA technology, we need a vector, right? We need a particular DNA fragment, right? Which need to be, or I should say the DNA fragment carrying gene of interest. So that need to be joined to the vector, right? So we need to cut the vector precisely, and we don't want the vector to be cut at random positions. We want it to be cut once, right? So for that, restriction endonucleases are used because they are very, very much specific. They recognize the specific base pair sequences in the DNA, and they cleave the DNA within the sequence. So here you can see the examples of restriction enzymes. Now, basically, when you talk about the classification of the restriction enzymes, so they are broadly classified into type 1, type 2, type 3, and type 4. Now, as we are not discussing about restriction enzymes here, so I will not go in detail. But yes, over 3000 different types of restriction enzymes has been discovered, and specifically type 2 restriction endonucleases are most commonly used in gene cloning and recombinant DNA technology. So here you can see the examples of type 2 restriction endonucleases. So EcoR1, BAMH1, here a few examples I have quoted. So EcoR1 isolated from Escherichia coli, and you can see this is the particular sequence it recognized. So they basically recognize the palindromic sequences, that is the sequences which are read same, that is which are read same if you read it from 5' to 3' on both the strands. So here you can see that EcoR1 is recognizing GAA-TTC, and specifically it is cleaving, like it cleaves, it recognizes its specific sequence and cleaves within it. Now, when the enzyme cuts, it can generate the sticky ends. So as you can see here, this is a sticky end, right, with an overhang region, right? And the another type of end that can be generated is the blunt end. So like for example, if you take this enzyme AGCT, so AGCT and the complementary strand, it will be T, C, G, and A, right? So when the DNA is cut at the same place in middle of the recognition site, like from here, the type of ends which are generated, they are the blunt ends. Unlike the sticky ends which have the overhang region, which you can see here, right? And they can stick back by transient base pairing. So sticky ends or cohesive ends, you can also call it as. Now, the restriction site where the enzyme cuts may be present anywhere in the genome. As every DNA is made up of various combinations of base pairs, a simple mathematical rule exists that can explain the probability of a given sequence. So for calculating the probability of a particular restriction site, it is based on certain fundamental rules, as you can see here. So these are the certain fundamental rules that you have to remember. So probability of finding a particular base at given position is always 1/4. This is a very important point that you all have to remember. Why it is 1/4? See, probability is what? It's a chance, right? So probability, like in mathematics also we calculate as the number of the favorable cases by the total outcomes, right? Like for example, if you flip a coin and I ask you that what is the probability of getting a head when you flip a coin once? So total outcomes are 2, because when you flip a coin, the coin will land either showing head or tail, right? So 2 outcomes are there. But specifically, what is the favorable case we are looking for? Let's say head. So probability will be 1/2. Similarly, here we are calculating the probability of finding a particular base at a given position, right? So it is 1/4 because there are total how many bases? There are total 4 bases. A, T, G, and C. So the probability of finding a particular base, let's say A, will be what? So favorable cases, the one that we are looking for, the event that we are looking for is A. But total outcomes are how many? So A, that is 1, right? And what is the total outcome? 4, because we can get A, T, G, C. These are the total 4 outcomes, right? So this is the reason the probability is 1/4. Now, purine or pyrimidine mentioned at a given position, like I'm sure you might have come across some recognition sequences where they have mentioned purine and pyrimidine as well in the sequence, right? So always the probability will be 1/2. So like for example, it is the sequence is mentioned like this, right? So purine is there. So purine can be adenine or guanine. Similarly, anywhere in a sequence, if it is mentioned pyrimidine, so it can be cytosine or thymine. So total outcomes are how many? Two outcomes are there, right? So this is the reason, like if you are looking for the probability of purine, so purines are guanine and cytosine, right? So probability of a guanine or a, now to find the purine or pyrimidine at a given position, the probability is always 1 by 2. Because when I say purine, so you all very well know that purine can be adenine or guanine, right? Similarly, the probability of finding a pyrimidine. So pyrimidine, you all know that it is cytosine or thymine in case of DNA, right? So in case of purine, total outcomes are 2. Similarly, in case of pyrimidine, total outcomes are 2. So if you're looking for the probability of, like if the purine is mentioned in a sequence, let's say the sequence is mentioned like this. So the purine here can be adenine or guanine, right? So any one base can be there, right? So this is the reason that the probability will be 1 by 2. So 2 here are the total outcomes. So for purine, it is adenine and guanine. Similarly, for pyrimidine, it is cytosine and thymine. Then if N is mentioned in a sequence, let's say it is mentioned like this. So N can be any nucleotide. And always remember, in case of probability of any nucleotide at a given position, the probability will always be 1, because any nucleotide means it can be adenine, guanine, thymine, or cytosine. And also remember that total probability of the whole sequence is the product of individual probability per position of base pair. So why I'm saying product? Because they are all independent events. Probability of getting an A is an independent event. Probability of getting a T is an independent event, right? So if you are writing if the recognition sequence is, let's say, GAATTC, so getting a G, probability of getting a G is an independent event. Probability of getting an A is an independent event. Like it doesn't depend upon that some other base, right? So it's an independent event. Then if again, then you are getting again A in a sequence, that is also an independent event. So this is the reason we will multiply, that is total probability of whole sequence. This 6 base pair sequence will be the product of individual probability per position of base pair. These are the some fundamental rules that you all always have to remember. Now, let's say in the exam, they'll give you some ambiguous or non-ambiguous sequence. So in that case, how will you determine the probability? Like I have told you some basic fundamental rules, right? But now let's understand these, now let's understand that how using these rules, like how will you apply these rules in finding the probability of a particular base in a non-ambiguous or ambiguous sequence. So when I say non-ambiguous, it means that none of the position has purine, pyrimidine, or any nucleotide. So the clear defined recognition sequence is mentioned to you. So let's say the example again, let me take GAATTC, right? So here you can see this 6 base pair. This is the 6 base pair recognition sequence. See, if you see the type 2 restriction endonuclease recognition sequence, they are either of 6 base pair or 4 base pair, right? So here in the 6 base pair recognition sequence, none of the bases, like none of the position of purine is mentioned, or pyrimidine is mentioned, or N is mentioned, that is any nucleotide is mentioned, right? So it's a non-ambiguous sequence. So always remember the probability of given restriction site is predicted as, so I already told you, the product of individual probability per position of base pair. So it is 1 by 4 raised to power n, where n is the number of base pairs in restriction site. So let's understand the details with the help of an example. So here you can see GAATTC. It's the non-ambiguous sequence because all the bases are well defined. So number of bases are 6, right? So it's a 6 base pair recognition sequence. So probability of finding so we use this formula 1 by 4 raised to power n, right? So n here is 6. So 1 by 4 raised to power n, that is 6, and that will give you 1 by 4096. So basically, why I have written 1 by 4 raised to power 6? Like 1 by 4 raised to power 6 means you have to multiply 1 by 4 6 times, right? So why 6 times we are multiplying? So this is the probability for getting G, this is the probability of getting A, this is the probability of getting A, this is the probability of getting T, again T, and then C. So this is the reason we are multiplying the individual probabilities, right? So as I mentioned, the total probability, we are calculating the total probability of whole sequence, that is product of individual probability per position of base pair. So on solving, you'll get 1 by 4096. So this indicates that 1 in every 4096 base pair, there will be a restriction site. Then what if the ambiguous DNA is given, right? So ambiguous DNA means that at least one or more positions they have in the recognition sequence as purines, pyrimidines, or N instead of any particular base. So probability of given restriction site can be calculated as 1 by 4 raised to power N into 1 by R. Now what is R here and what is N here? So N is the number of non-ambiguous bases in restriction site and R is the probability at ambiguous base. Now, please note that for purine and pyrimidine, the value of R will be always 2, as I explained earlier. And for N, the value of R will be always 1. So further, to understand it in detail, let's consider an example. So this is a sequence G, T, N, and you can see, so N, any nucleotide. N means any nucleotide. And then pyrimidine, so it can be cytosine or thymine. Then again, you have thymine and then cytosine, right? So here, well-defined bases are 4, G, T, and C, right? But what is the value of probability of N? We all know 1. And what is the probability of pyrimidine? So it is always 1 by 2. So we all very well know the formula 1 by 4 raised to power N into 1 by R. So that is 1 by 4 raised to power 4 into, for N it is 1 by 1 because R value is 1 here, and 1 by 2 because R value is 2 here. And on solving, you'll get 1 by 512. Now, let's check some other formulas as well, which are very much important from exam point of view. What if in the exam, they even give you the genome size as well? So always remember, if in the question they mention about the, let's say, bacteria they're talking about. So bacterial genome they mention. What is the probability of getting a 6 base pair recognition sequence in 6 into 10 raised to power 6 base pair genome of Escherichia coli? So in that case, the formula that you have to use is number of possible site equals to size of genome or vector or DNA. Like in the question, they can talk about vector also, they can give DNA also, into P. So P is the probability, and that too in fractions you have to write. Alternatively, another formula that you can use is, so number of possible sites in given DNA, that is X, is size of the given DNA by 4 raised to power N into R. So where you all very well know N is the number of the bases in the site. So let's see what type of questions are asked in the exam. So this is one of the questions I've taken from previous year question paper. So genomic DNA isolated from a bacterium was digested with restriction enzyme that recognizes a 6 base pair sequence. Assuming random distribution of bases, the average length of fragments generated is. So here in this particular question, they have not mentioned the genome size of the bacterium, right? It is only mentioned that restriction enzyme recognizes 6 base pair sequence, and even the sequence they have not mentioned. So we very well know that in order to determine the probability of a restriction site, we use the formula P equals to, P is the probability, that is 1 by 4 raised to power N, right? So when formula we are using P is equals to 1 by 4 raised to power N, so N here is 6. So you very well know it will be 1 by 4 raised to power 6, and on solving you will get 1 by 4096. And it is very well mentioned, like you will not write the answer 1 by 4096, you will mention 4096. Why? Because here we have to determine the average length of the fragment that will be generated. So the point is that the enzyme will cut once after every 4096 base pair. So definitely, so if this is the, like here, this is 4096, right? Base pair, so enzyme will cut here. Then again, after 4096 base pair, it will cut. So this is the reason the answer you will be writing as 4096. Let's check another question. So here you can see the another question. So this is another practice question. Determine probability of finding a site of ALU1 restriction enzyme. So you can see 4 base pair it is, right? In a bacterial genome of size 6 into 10 raised to power 6 base pair. So here you can see even the bacterial genome size is mentioned. So here, first of all, you will read the question carefully, and then you will note down what all values are given to you in the question. So restriction site, 4 base pair mentioned. Probability of finding this site is, you know the formula for probability, it will be 1 by 4 raised to power n, because it's a non-ambiguous sequence, right? So directly 1 by 4 raised to power n, where n here is the 4, right? And so on solving, you will get 1 by 256. Now, this is not the answer, right? We have to solve the second part of the question as well, because we have to keep in mind that the size of the bacterial genome is also mentioned. So here we'll be using the formula number of sites in given genome, that is genome size into P, that is probability. So this is the particular formula we are using here, right? This one. So if vector would have been given, or any other DNA fragment, like if DNA fragment would have been mentioned, so accordingly, like the formula remains the same. So genome size into P, so it is 6 into 10 raised to power 6 by 256, and on solving you will get 2.34 into 10 raised to power 4. That is 23,400 sites of ALU1 present in the bacterial genome. So I'm sure now you understood how to solve such type of questions, that is the questions based on probability of finding a restriction site, right? Now, it's very easy when the nucleotide composition is same throughout the DNA. So probability of finding a base will always be equal, right? But what if they mention that the GC content is 60% or 30%? So in that case, in such questions where they mention that probability in like they mention probability in GC rich or GC poor DNA. So in that case, when the DNA sequence is biased for specific type of base pair composition, where you have GC more or GC less, then in that case the standard rules for probability would change. Always remember this point. So bias sequence in favor of GC or AT would be considered. How? Let's understand it with an example. So if GC content is given 60%, so probability of finding G or C. So 60% is total GC content. So that means G is 30%, C is 30%, right? And or I can write 1 by 3.3. Say, and if the GC content is 60, like out of 100, GC content is 60, then let's say they mention that the probability of like 80 they mention is 20%, right? So like 20% how? So if GC content is 60%, that means the remaining 40% is your AT content, right? So adenine, so further adenine 20 and thymine 20, right? So probability of finding A or T is 20%, that is 1 by 5. So as per conventional rule, this is how, like as per the conventional rule, overall probability of the site will be the product of probability of individual base of the site. So these are some key points that you all have to remember while preparing this particular topic. So this is all for now. I am sure today you have learned something really important and something really new. So all the very best. Don't forget to ask any questions if you have. You can ask your questions. You can post your queries in chat support of our in our Biotechnica website. And all the very best. We'll see you in the next video. Do like, share, and subscribe our channel. Take care and thank you.